Hi, this is Maheshwari D, and now I'm going to explain about polymerase chain reaction. Polymerization chain reaction is a laboratory technique for rapidly producing millions to billions of copies of a specific segment of DNA, which can be studied in greater detail. PCR involves using short synthetic DNA fragments called primers to select a segment of genome to be amplified and then multiple rounds of DNA synthesis to amplify that segment. PCR tests are used to diagnose certain infectious diseases, identify a genetic change that can cause disease, find small amount of cancer cells that might be missed in other type of tests. Working of PCR: taking a sample of blood, saliva, mucus, or tissue. The sample will contain your own DNA and possibly the DNA of a pathogen or cancer cell. The sample is put in a specific machine and analyzed. An enzyme called polymerase is added to sample. This causes the sample to produce copies. The copying process is repeated multiple times. After about an hour, billion of copies are made. If a virus or a pathogen is present, it will be indicated on the machine. Certain viruses, including COVID-19, are made up of RNA rather than DNA. For these viruses, the RNA must be changed into DNA before copying. This process is called reverse transcription PCR, RT-PCR. PCR and RT-PCR check for the presence of pathogen. Another type of PCR known as the quantitative PCR, qPCR, measures the amount of pathogens in the sample. qPCR can be done at the same time as PCR or RT-PCR. Steps involved during PCR: denaturation. Heat the reaction strongly to separate or denature the DNA strands. This provides single-strand template for the next step. Annealing. Cooling the reaction so the primers can bind to their complementary sequences on the single-strand template DNA. Extinctions. Raise the reaction temperatures so tau polymerase extends the primary synthesis new strands of DNA. This cycle repeats 20 to 35 times in technical PCR reactions, which generally takes 2 to 4 hours depending on the length of the DNA region being copied. If the reaction is efficient, the target region can go from one or few copies to billions. Now you can see how a single DNA can be copied into millions and billions of copies. Limitations of PCR and RT-PCR: the PCR reaction starts to generate copies of target sequence exponentially. Only during the exponential phase of the PCR reaction it is possible to explorate back to determine the starting quantity of the target sequence contained in the sample. Because of inhibitors of polymerase reaction found in the sample, reagent limitation, accumulation of pyrophosphate molecules, and self-annealing of the accumulating product, the PCR reaction eventually ceases to amplify target sequences at an exponential rate and a plateau effect occurs, making the endpoint qualification of PCR products unreliable. This is the attribute of PCR that make real-time quantitative RT-PCR so necessary. Thank you.